Thank you. I'd like to start out by thanking Gully for a very nice introductory talk. I feel like if we went through that slide by slide, they could all stimulate interesting conversations in the discussion section. So I'm going to talk to you today about a corpus linguist perspective on a corpus called the Colorado Richly Annotated Full Text Corpus. I'll explain more about what its contents are. So most of you know me as a natural language processing person. I also work as a corpus linguist. And when we do corpus linguistics is the science of building annotated text collections and using them for linguistic or natural language processing research. And building corpora always involves making mistakes, cutting corners, and other miscarriages of intent that end up affecting the final product. So what I'm going to talk about today is this corpus from the view of a corpus linguist with an emphasis on lessons that we learned from the process of building it and things that I wish we'd do differently and that I suggest that you do differently in your future corpus building efforts. So the Colorado Richly Annotated Full Text Corpus, which I'll refer to as CRAFT, is a collection of full text journal articles annotated with linguistic structure and semantic concepts. And the motivation for building something like this is threefold. So first of all, there's a necessity for high recall information extraction. And one thing that's been very clear for a long time is that if you want to have high recall information extraction, you can't just work with abstracts like most people in biomedical natural language processing have for many years. You really need to work with full text journal articles. Another is the necessity for improving tool performance. So most of the tools that we use in biomedical NLP today were built using abstracts as their training materials and evaluation materials. And one thing that we found as full text articles have become more available and we've been able to evaluate those tools on the full text articles is that tools built for abstracts don't work as well for full text. And finally, there's the increasing availability of full text. So this is something that's really new for us in biomedical natural language processing, and it's very clear that there's a big resource out there that we could be tapping if we know how to do it. So that was the motivation for building the corpus. How it's relevant to biocuration is that this is currently the best resource for building natural language processing tools for detecting the kinds of stuff that most of us who are doing, or most of you who are doing model organism database curation care about. So gene ontology concepts, Entrez gene IDs, phenotypes, and other things that you might be interested in. So our approach to doing this is what was called a multimodel annotation task. And what that means is that we use different kinds of people and different task definitions for different kinds of data that we wanted to annotate. So we annotated two kinds of things, linguistic structure and semantic concepts. So to do the linguistic structure, we use linguists, mostly graduate students, to annotate the semantic concepts, which we're defining as things that are present in some ontology. We use domain experts from mouse genome informatics and from the University of Colorado School of Medicine. The second annotation project is sometimes called a light annotation task because it doesn't require insight into linguistic analysis. So the materials that we used, the way that we sampled the full text document collections out there are that every document that we selected was used as evidence for at least one mouse genome informatics gene ontology or mammalian phenotype ontology annotation. So first of all, it had to have been used by MGI. Second of all, it had to be available in open access in the PubMed Central XML format. So this is an XML format that PubMed Central makes available for distributing their documents. It's very clear how to parse it, how to find sections, and so on. The exclusion criteria is basically that you didn't meet the inclusion criteria, so we had one document that was available only in abstract form. So what this led us to was a total of 97 full text documents comprising a little bit less than 600,000 words in size, which is bigger than almost every biomedical corpus out there. And there are a couple of questions that you could ask about this. So two things that we worry about in building corpora are they balanced and are they representative? So the notion of balance has to do with sampling the space of data in some principled way that spreads you across whatever kinds of variables are going to be of interest. So that might be genre, it might be authorship, it might be journals in which you're published. And 97 articles are probably not balanced. Representativeness has to do with whether or not the document collection represents the full range of variability of kinds of ways that things might be expressed in language. And this probably is a pretty representative collection. So it covers development, physiology, genomics, disease, all the kinds of things that MGI annotates, which, as you know from our previous talk, is quite a bit. So I want to talk a little bit about the annotation model. So annotation projects, implicitly or explicitly, are based on a model of annotation. And we can describe this model as a triple of a vocabulary of terms, the relationships between the terms, and the interpretation of the terms themselves. This idea is due to Pustiavsky and Stubbs. And so I'll talk about what we did in terms of the annotation models. On a high level, this is what we call a multi-model annotation task. So what that means is that we had two different models that we used, one for the linguistic annotation and one for the semantic annotation. And so for the linguistic annotation, we did sentence boundaries tokenization, which is splitting the input into its parts. So a token might be a word, it might be a punctuation symbol, it might be the constituent parts of a hyphenated word. These are kind of the building blocks of the document collection. We did part of speech tagging, analyzed syntactic structure, and also did co-reference, which has to do with finding when two things in a text refer to the same thing. So if I said BRCA1 and then later said this gene, co-references that shared meaning between the two. And we also marked up discourse structure, and in particular how arguments were laid out in the structure of these documents. So the semantic annotation consisted of two things. So one is various open OBO or Open Biomedical Ontologies Project ontologies. So semantic concept in this case means it exists in some OBO ontology, and then Entrez gene identifiers. So most of you are familiar with Entrez gene. It's the big NCBI database of genes that has, I don't know how many millions of entries at this point across many different species, each with a unique identifier. So to look a little bit more closely at the linguistic annotation model, so if T is the terms, then the terms in this case are things like part of speech, syntactic structure, co-reference chain, and so on. The relationships between them are that part of speech can be seen as NN means a noun, NNS means a plural noun, and then P is a proper noun, and so on and so forth. And so there's a set of about 40 of these that we used. And if you're wondering how you get from the 8 parts of speech that you learned in grade school to 40 parts of speech that are used in natural language processing, it's by subdividing the categories that you learned in school into finer and finer grained classes. And then the interpretation of these is that part of speech is a list of what we call lexical categories. A base noun phrase is a noun and its left word, identifiers, and so on. So the underlying theory behind the annotation model is that we wanted to avoid creating a new standard. So there are many, many standards for doing annotation, and we didn't want to add a new one. We wanted to try to help some pre-existing one gain traction by adding on a new use to it in this domain. So So it's my belief that picking a pre-existing standard is sort of a political issue, and some of the ones that went into what we chose basically were trying to predict what projects look to us like they're going to be successful and glomming onto those. So one that we adopted from pretty freely is the Annotation Project, which is a very, very large multi-center, multilingual annotation project. We had to make some adaptations to those standards for this domain. We had to add some new part-of-speech tags. We had to add some new phrasal categories. And in the definition of discourse categories that we use, some of them we had to omit, some of them we had to collapse. So unfortunately, we weren't able to use the standards quite as they were designed, but we stayed as close to them as we could. This underlying theory of not creating a new standard got us into a little bit of trouble. So one example of that is that the fully defined co-reference annotation standards can't be shared. So the OntoNotes co-reference annotation standards that we used are made by BBN, and they're under copyright. So BBN has published a paper about their annotation standards that lays them out pretty closely to the guidelines, but the guidelines themselves they're not willing to release. And that's a really big problem. If you do an annotation project, you really want to be able to release your annotation guidelines along with your data. The annotation model for the semantic categories, the terms were semantic concept, first of all, and gene ontology term, cell ontology term, and so on and so forth. We used 6 different ontologies for this. So Go, the cell type ontology, KEBI, the NCBI taxonomy, the protein ontology, sequence ontology, and Entrez genes entry. And an ontologist would really chew me out for having used the word term and would very much prefer to have the term concept in here rather than term. The relations for this were that a semantic concept could be a gene ontology concept or a cell type ontology concept and so on. And then the interpretations of these are that a gene ontology term or a concept is a list of all the concepts in the gene ontology and so on. So you'll notice that this is very, 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 very ontology centric. The only non-ontology we have in here is Entrez gene. So the underlying theory behind the semantic annotation model is that only ontologists really understand how to annotate semantics. And this got us into a lot of trouble. So one was a weakness in the general annotation model. So one of the principles here was if it's not in an ontology, it doesn't exist. And this is a problem. So one of the basic principles of doing annotation is you always need to give the annotators a way out. If there's something that they think they should be annotating, but they're not quite sure what they should do with that, you need to give them a way to mark that up so that you can figure out how to integrate that into your general annotation schema. And this led to a very time-consuming complete second pass through the entire corpus after all of the other annotation was done to annotate the protein families. So I mentioned that we used Pro. We did not use it on our first pass. We used it as the second pass when we sat down and looked at the final project, and there was general recognition of the fact that because if the annotators were not able to map a gene mention to an Entrez gene identifier, either because it was unclear what the organism was or because the author was being deliberately unclear about, say, wanting to use a term to refer to an Aachen gene, and so they didn't want to be specific about what the organism was, or couldn't be specific about what the organism was, we just didn't annotate it. So we had all these unannotated references to genes. And so we ended up making this whole second pass, and we really could have anticipated this by seeing the weakness in this idea that if it's not in an ontology, it doesn't exist. And then So by going at this with the assumption that only ontologists know how to do annotation led to general inefficiency of effort and poor use of limited personnel resources that could have been avoided with more input from linguists and people who understand how to do annotation in general into the process. So the annotation processes for the linguistics and the semantic annotation I'd like to talk about a little bit. So for the linguistic annotation, the annotators had 1 year of master's level coursework in linguistics at a minimum. They needed to have previous experience with syntactic tree banking and required 6 months of training to be able to do this work. What they annotated to do the sentence segmentation, and sentence segmentation means splitting your input into sentences. That might sound like a really, really easy task. It's much, much harder to do than you would think it is, actually. And so the sentence segmentation, the tokenization, which is splitting up into parts, words and punctuation and so on, and the tagging with parts of speech was done automatically and then manually corrected. The syntactic parsing, or the analysis of the syntactic relationships between the phrases and the data, was done automatically and also, again, manually corrected. The addition of node labels that were not in the Penn tree bank set and the addition of function tags, I can explain what those are in the question session if there's interest, was fully manual, and the co-reference annotation was fully manual. And I'll talk about why I wish I'd done that differently in a minute. The annotation project for the semantic part of this, the annotators were 3 people with PhDs in the biological sciences, including one of the MGI curators. And we did an automatic pass for exact matches, plurals, and exact synonyms, and then followed that up with a very intensive manual pass. We did 1 ontology at a time, so this is something that I think we did right. A general principle of how to approach an annotation task is pick one thing, preferably the easy thing, do that through the entire corpus, and then make consecutive passes for the other tasks that you need to do. This was single blind annotation by one primary annotator, which was then corrected by a senior annotator. And as I'll discuss in a minute, I think this was something that we did wrong. And the senior annotator then had weekly telephone meetings and consensus construction of the data set where he disagreed with the primary annotators about the work they had done that week. You'll see how many weeks this was in some of the slides that I'll show you. What I wish we'd done differently in here is a few things, actually. So for the co-reference annotation, so co-reference is generally between some kind of syntactic unit, some kind of grouping of words and phrases. And so you'd like to do this with a syntactically annotated group of documents. We didn't have one because the co-reference annotation was happening at the same time that people were doing that manual correction of the syntactic analysis that we had produced automatically. So I just had, in this case, the annotators pick out their own syntactic units to make co-refer to each other. And in retrospect, it would have been really easy to have built an automatic base noun phrase chunker, that being the unit that we were using in the co-reference annotation, and let the annotators correct that when and only when it was necessary for co-reference, and then thrown out that automatic annotation afterwards. We should have used more semantic annotators. The approach to this, as I said, was that we had single annotators working on the entire document set for whatever task they were working on, and one senior annotator corrected everything. And what would have worked much better would have been to train the annotators, figure out the inter-annotator agreement at the beginning, get it to a high level, and then split the work and do less of this double annotation. So probably more single blind annotation, frankly, probably would have been much more efficient. And if we had good inter-annotator agreement from the beginning, just as effective as what we did, and it would have taken much less time and cost us a lot less money. And finally, the guideline development was done with a single annotator, and it would have been much more effective, I think, to involve multiple annotators. As it worked out, I think we had a pretty good set of guidelines for this based on the fact that multiple annotators were able to apply them with higher inter-annotator agreement, but we probably could have gotten those guidelines more quickly if we had involved multiple annotators in that process. So I want to talk a little bit about evaluation and quality control, and I'll come back to the notion of inter-annotator agreement in the course of this part of the talk. So the quality control for the tree bank, or the syntactic analysis, was first of all manual resolution of disagreements between the annotators by the lead annotator. And then we used a software tool called Corpus Search that did about 150 checks for things that should never occur in the syntactic trees. So for example, in the first example that I've given you there, PP means a prepositional phrase. You notice there's no preposition in there. So that clearly should not have been tagged as a prepositional phrase. In the second example that we have a sentence level node and a verb phrase level node at the same level in the hierarchy, and all you need to know about this is that that should never happen. A verb phrase can be a daughter to a sentence node, but they should never be at an equal level. I can talk about the details of what that means later if you like during the questions. And so we had about 150 rules that checked for this kind of violation of things that we knew should never happen in a corpus. There were still bugs that snuck through this. So one of the symptoms of this is that to use a corpus for linguistic research, you want to be able to search the syntactic trees with a tool called tgrep. To run tgrep, you need to prepare your corpus with a tool called tprep, and the tprep corpus, for reasons we still haven't figured out, will not work on our data set. So although we did put a lot of effort into the quality control for the tree banking, it's clear that some errors snuck through there. So one of the things that we use to judge the quality of any annotation guidelines and of the eventual annotation effort is something called inter-annotator agreement. And this is an idea that ultimately goes back to psychology. And the question is, if you've got some non-observable that you want, judgment that you want to make about something, how do you know if you're doing it correctly? And the idea is that if multiple people see the same thing in the absence of that overt signal, then it's probably correct. So you want high number, so when we measure that agreement between annotators, we want that number to be high. So for the linguistic annotation, oh my. Sorry? Something terribly wrong with my figures here. For the linguistic annotation, basically the bottom line is we had quite high numbers, so numbers above 0.9. If you're doing above 0.8, you're probably doing pretty well, and we had numbers above 0.9 for pretty much everything. So for semantic annotation, I had a couple of slides with beautiful charts here to show you. What they look like was they started out with very low annotation, which climbed relatively quickly and had a very spiky appearance. So one of the things that we noticed was that as we came to new articles, often there was a new set of semantic concepts that would throw off the annotators, and so we would have these dips in the inter-annotator agreement periodically, which would then climb up as we moved on to easier topics. And so what would have been nice would have been to have reached that high inter-annotator agreement earlier in the process and then done more single blind annotation than we did. So what we got out of this is about close to 100,000 semantic annotations that included about 4,300 unique semantic concepts. We have about 600,000 words that are linguistically annotated, including syntactic structure. We have co-reference and discourse annotation, but haven't released that yet. This is fully available for free with a very unrestricted license, so you can do anything you want to with it. The current version is 1.0. Somewhere between there and version 3.2, we'll add in two subsets of the corpus that we held out for shared tasks. So the data set was just released late last year and has already been used for 3 experiments that we know of. And finally, some acknowledgements to the people involved, and I have 26 seconds left to go, so there's plenty of time for questions. So maybe with my 17 seconds related, I'll point out that if biocreative is not leading to useful tools, we're really failing to do what we do. So that's why we do biocreative. As compared to other competitions, they focus on linguistics. Biocreative tries to focus on being useful. And regarding ProMiner, not only does it participate in biocreative, but it came from biocreative as a product of the first biocreative. So you see that we can produce things that are useful. I'll stop now.
I have 2 questions. The first one is how long it took the whole process of creating this corpus?
4 years.
4 years?
4 years.
Okay. And the second one is, did you do any processing of after you run with the different ontologies of overlapping? Because sometimes like you will have overlapping ontologies in a text and any context that you could actually facilitate curators instead of looking at a lot of false positives.
So are you talking about cross products between the different ontologies?
No. I'm talking, for example, simple things. Let's say you have, I'm trying to think of something. So let's say TGF-beta signaling pathway, it's a process in Go.
Yes.
But TGF-beta could be a protein, right?
Yes.
So you have an overlap in there
Yes.
of detection. Do you do any processing there?
Yes, we did annotate those. So TGF-beta signal processing is annotated to Go in that case, and then TGF-beta is annotated to Entrez gene.
But my question is, I would not annotate TGF-beta to a protein there because the whole thing
Well,
might
they
be just
overlap.
the process.
They overlap.
My question, okay.
Any other questions? Oh, Max.
Very quick, how many articles did you annotate, or maybe I've missed it, sorry?
97. So that was the number of articles that MGI had used as evidence and that were also in the PubMed Central Open Access subset.
Did you select them randomly? Were there some?
We took every one there was. There were not that many. Because there were only 97, we used all of them.
Oh, okay. And do you know how much you cover the gene namespace? How many genes roughly were there mentioned in there?
We do know, I don't know the number off the top of my head.
Okay, thanks.
Yeah, sure.
Any more questions? So thanks to the speaker again. That was a terrific talk. Thank you. So that concludes the first session.